Hi everyone, my name is Deepika, and the question that I'm trying to address through this poster is what is the origin of slow solvation dynamics in DNA? Now, historically, it was considered that only biomolecular structures perform all the biological functions, whereas water remains as a passive matrix. However, now it is well known that water molecules are crucial for any biomolecular structure to perform its function, and they are not mere spectators. Experimental endeavors on this dynamics, probed by time-resolved fluorescence Stokes experiments, have shown that such collective dynamics varies from few picoseconds to nanoseconds, but the origin of such dispersed dynamics is not understood. In fact, recent MD simulations have tried to address this issue, however, they were not long enough to capture the slow dynamics. But the question arises, why does it remain so difficult to understand? That question can be answered through this plausible answer that it is due to the complex electrostatic coupling between the large ionic DNA and the surrounding water molecules and ions. And this involves the solvation response from different length scale. So what we have done here is we have taken a minor group binder that goes and binds to the ETRH minor group of well-known recursion DNA. We confirm the binding using CD spectrum as well as the steady state spectrum, and we measure the binding constant, which is very high, that is of the order of 10 to the power 8. So it is impossible to capture the spot dynamics using one single experimental technique. So what we did is we calculated the wavelength dependent decays using two experimental techniques, time related single photon counting and fluorescence subconversion, and we constructed time resolved emission spectrum from 100 femtoseconds to 10 nanoseconds, and then we calculate the absolute Stokes shift. So unlike pure water, the dynamics doesn't finish within few picoseconds, but rather continues till nanoseconds. Now, why does it happen? To find out this, what we did, we carried out is 800 nanoseconds of long simulation. In fact, this is the longest simulation that has been used to calculate the solvation response or the solvation correlation function. So post-running simulation, what we calculate is the interaction of this probe DAPI with all the atoms of my system. So what we get is the electrostatic energy fluctuations of the system, which are represented in black. Blue represents the electrostatic energy fluctuations of water. Green represents fluctuations of DNA. Red represents fluctuations of ion, sodium plus ions. So if you carefully look at these fluctuations, you see there is a strong coupling between them, and there is a large anti-correlation between iron and water and DNA and water. Now, when we directly compare our experimental and simulation results, what we see is that beyond 2 picoseconds, beyond nearly 2 picoseconds, the time correlation function calculated using MD simulation is capturing the essential dynamics as seen in the experiment. But to find out the origin, we decompose this total into individual components. So this is my total correlation function, and this is correlation function of my DNA water, and this is correlation function of my DNA. So what we see is that the dynamics is mainly controlled by the correlation function coupling of DNA and water. Now to find out whether it is really so, what we did is we froze down the DNA fluctuations and keeping other atoms free. So what we see is that on freezing DNA fluctuations, the long-term dynamics beyond 100 picosecond completely eliminates. So this means that the coupling of water and DNA was giving rise to this long-term dynamics. But to really address this issue quantitatively, we did froze down the water fluctuations. And what we see is that the long-term dynamics completely eliminates. In one single snapshot, it finishes. So this means that water fluctuations are indeed important for any biomolecular structure to perform its function. So to conclude, the origin of this slow dynamics lies in the slow fluctuations of DNA parts which perturb the nearby water and ions to give rise to the slow solvation dynamics in the Dickerson DNA. Now, what is the biological significance of such a dispersed dynamics is that these fluctuations, this coupling of water and DNA motions, is really important for mismatched DNA, where this coupling generates mismatched specific signatures that give signals to the repair enzymes to come and repair it. And I'm happy to share that this work has been published in Journal of Physical Chemistry B as a cover page article. Thank you.